So before I get into how we work with Covaris, just a quick introduction to IQ Proteomics. We've been around for almost 10 years now. We have a lab in Framingham, Massachusetts, right across from Jack's Abbey Brewing, if you know where that is. It's about 30 minutes west of Boston. We're a full-service proteomics laboratory focusing on quantitative proteomics, initially founded on isobaric proteomics multiplex workflows. That's what the I stands for in IQ. But we've moved a lot more into targeted workflows as we've matured as a company, and that encompasses a lot of what we do these days as well. So we have 5 mass specs in the lab currently, a dedicated informatics pipeline, and all the work's done at our laboratory in Framingham. So as a CRO, one of the interesting things about working with a lot of different companies is that it's really important for us to be super flexible in what sort of samples we can handle, and we really pride ourselves in being able to work with a really wide breadth of sample types, sample input amounts, lysis buffer requirements, just really being an extension of our customers' laboratories. And as you can imagine, every company has a slightly different workflow. Some companies have quite a bit different workflows, and whatever we can do to be more flexible with our sample prep workflows and in terms of our internal capabilities just facilitates our ability to provide high quality data sets in a rapid timeline, which is what everybody wants. And so one of the things we think about a lot is lysis, sample lysis. That's the first step in every workflow. There's a number of criteria we look at when we're evaluating what to use for a particular project. Obviously we need to be compatible with broad breadth of samples, like I just mentioned. We want to be able to deal with viscosity associated with lysing the nucleus, high recovery with low input amounts, compatibility with 96-well plates, and then that leads into integration with automation. So we use bead beating a lot in the lab, historically and currently. That's great for any sample you want to throw at it pretty much. Does a good job of shearing DNA. That's not great for small sample types. We haven't found a way to get that into 96-well plates, and there's no way that we know of to automate that. Just mechanical lysis by pipetting works really well for small sample types. It's very compatible with 96-well plates and robotics, but not necessarily compatible with anything other than cell pellets. And some lysis buffers are compatible with benzonase and other nucleases for getting rid of the DNA in the sample, but some customers like guanidine, for example, and we can't use those reagents to get rid of the viscosity. So sonication really solves all of those problems, especially the implementation of it that we have, which is the R230 from Covaris. We can throw pretty much any sample at it. Does a really good job of shearing and cleaving DNA. Works well with small samples and large samples, as a matter of fact. It's just natively is able to use 96-well plates, and it integrates well with our robotic sample handling. So we use this R230 focus ultrasonicator from Covaris. There's a lot of technical details on this slide, but the key points are 96-well plates, temperature-controlled sonication, and native integration with automation that sits on the deck. So we've used a lot of different consumables that Covaris offers, and we've really settled on this microtube snap cap bead glass vial for our lysis of most of our samples. Holds about 120 microliters. Sits in this plate manifold that is compatible with the Covaris R230. We've done anything from tens of thousands of cells in this thing to up to over 5 million cells, as well as mammalian tissues. So easy ones like liver and spleen to more hard ones like heart, for example. You have to extend the sonication time a little bit longer for some of those harder tissues, but it works really well for almost everything. Covaris has some protocols for formalin-fixed tissues in this thing too. We haven't done that yet, but apparently that's also something that works really well. So I mentioned integration with automation. So this is our implementation at IQ Proteomics. We've got a TCAN Fluent that we've got integrated directly with our R230. You can see it at the back there. Also, right by the R230 there is a little hole in the deck so that the robot can reach down into a UV-vis spectrometer to do BCA measurements. We've got a magnetic station for doing bead enrichment workflows, and we've got about a dozen hotels there for temperature-controlled digestion and incubation. So automated sample prep at IQP looks something like this. This is plate-based workflow, starting with a sample plate of, in this case, cell pellets that might have variable amounts in them due to just inability of pipetting to get accurate cell amounts in each well, or experimental details that might cause different growth in the wells of the experiment. We add lysis buffer and put that in the Covaris for lysis. That gets directly handled by the TCAN for a BCA assay. The TCAN then automatically does normalization of concentrations and input amounts for arraying a new plate for downstream work. It adds ceramag beads and then some ethanol for the precipitation to start an SP3 workflow. So the advantages of this workflow in our hands are obviously the throughput, hundreds of samples a day compared to dozens manually with far less hands-on time. Very high yielding process since we're starting in plates right from the beginning, and introducing robotics earlier in the workflow dramatically improves reproducibility and scalability. So just a quick little data vignette about that. So this is we've compared bead beading of 16 replicates of 1 million HeLa cells compared to lysis on the Covaris. You can pretty easily see that we've got quite a bit higher yield on the Covaris, but what's maybe a little bit harder to see is that our reproducibility is quite a bit better. This is important for us, in particular for smaller sample amounts where we're often limited to the lowest yielding sample in whatever workflow we're doing, and improving the variability across lysis of hundreds of cells just allows us to be able to produce more robust data sets for the customer over a large number of samples. So I mentioned that we also do downstream magnetic bead workflows for our digestion. So we've also got that automated on the Covaris integrated with the TCAN. So essentially what we're doing there is using the TCAN and the Covaris to hand back and forth the plate. TCAN's adding and removing wash buffer, incubating the plate. The Covaris is agitating the beads and resuspending them during the washes. And then that cycle is repeated again when we're doing the elution of after we've digested the samples. We go do a few cycles of elution and agitation and washing the beads in the Covaris. So outcomes eluted peptides from that are ready for downstream, either TMT labeling or direct LC-MS acquisition for data-dependent workflows. So this is just a quick comparison here of how this works in our hands compared to what we consider to be the gold standard, which is methanol chloroform precipitation. Comparing 20 micrograms of input either by methanol chloroform on the left to different amounts of input on the right with SP3 automated on the Covaris. You can see over a pretty wide range of sample input amounts we're generating, and sorry, I should say that we're loading 1 microgram of input equivalent on our mass spec for I think a 45-minute LC run, comparing how many peptides we're able to identify for each of these experiments. Over a wide range of input amounts, we're competitive or actually better in terms of peptide IDs than the gold standard methanol chloroform. Reproducibility across the replicates, as seen by these error bars, is also improved compared to the methanol chloroform workflow. And this has just been a game changer for us to increase our throughput and improve our variability and just get better data for our customers. So I think I'm a little bit early here, but just in summary, Covaris R230 and its integration with our TCAN instrument has just really allowed us to unlock high throughput sample prep for a variety of our workflows for our customers and really just get good quality data in the hands of our clients as fast as possible. And what that looks like for IQP is global proteome dynamics, chemoproteomics, in particular covalent inhibitor screening is a huge aspect of our work, and then a number of targeted workflows as well. So I'd just like to thank most people at IQ Proteomics, but in particular, Brian Erickson, Ryan Coons, Allison Erickson, Kurt Baltier, Rohit Avani, and Brian Sukovich, Covaris, the whole Covaris team, but our main contact there has been Samir, who's really helped us push through a lot of these optimization of these workflows for us, and just happy to answer any questions.